Welcome to Zoology Classroom. Today we are going to know about sperm sexing. Sperm sexing, this topic comes under Animal Biotechnology paper in MSc Zoology 4th semester. Let us know what is sperm sexing. Sperm sexing is a technology. This technology is also called as sexed semen or sex sorting or sperm sexing technology. This is a very new technology. Mostly this technology is being used in dairy industry. And this technology was developed by an agency called as USDS, United States Department of Agriculture, a government department of America. And this is located in Livermore of California and another location also there. It is located in another place that is Bellsville in Maryland. So, and this technology was patented as Bellsville sperm technology, sperm sexing technology, and it was commercialized in 2001 in America. So coming to definition of sperm sexing. So we can define sperm sexing as the semen which has been selected based on its capability to influence the gender of the fetus. Technically, it can be defined as semen having X or Y bearing sperm to produce progenies of a desired sex, either male or female, with almost 80 to 90% accuracy. So sperm sexing is a selective technology. Usually we don't know in case of born children or young babies, whether it could be a male or female. It will be purely depending on luck factor. But in this sperm sexing technology, wantedly, we would like to have either male offspring or female offspring. So for that purpose, we have to select the sperm. Either it will be having X chromosome or Y chromosome. We know that sperms having X chromosome, when they are united with ova, they will give rise to a female baby. And sperm having Y chromosome, if they are united with ova, they will give rise to a male baby. So in this way, the sex of the baby or sex of the offspring is directly controlled by the sex chromosome of sperm. So that's why we know that the sperms, half of the sperms will be having X chromosome, half of the sperms will be having Y chromosome in any semen sample. So in any semen sample that we take, 50% sperms will be having X chromosome, 50% sperms will be having Y chromosome. So, but, and the number of sperms also will be in millions. So million number of, n number of, many number of sperms will be there. Out of those millions sperms, only one sperm will be succeeded in entering into the ova. So, and which one enters into the ova, whether it is a Y bearing sperm or X bearing sperm, we don't know. It is not in our hand. It is purely decided by the luck factor. But here we don't want to leave this to the luck factor. We want to control. So which chromosome bearing sperm has to be united with ova. So we want to, the scientists wanted to control that thing. For that purpose, they wanted to separate the semen into two parts. One part will be having X chromosome semen, another part will be having Y chromosome semen. If we want to have a male offspring, then we will use semen having Y chromosomes only. If we want to have female offspring, then we will be using only X chromosome bearing sperms only. So for that purpose, it should be in our hand. For that purpose, they wanted to invent a technology, and these American scientists, they were succeeded in devising a technology, and we are calling that as sperm sexing technology. So, and what is the principle? So, on what principle? What is the basis for this separation or sexing or separating the sperms into two types: X-bearing X-bearing sperms and Y-bearing sperms. So, what is the basis for this thing? So, we know that Y chromosome and X chromosome are different. Y chromosome is longer than X chromosome. So there is a difference in between X chromosome and Y chromosome in terms of size, and that reflects in terms of DNA content. So Y chromosome will be having more DNA than, X chromosome will be having more DNA than Y chromosome. So this is the basis for separating sperms into two types. So this is the basis for this sperm sexing technology. Usually the difference between these X chromosome and Y chromosome will be different to each species. In case of humans, X chromosomes will be having 2.9% of additional DNA content than Y chromosomes. Whereas in cattle, the X chromosomes will be having 3.8% more DNA than Y chromosome. So this is the basis for separating sperms into X chromosome sperms and Y chromosome sperms. So this is the basis for this sperm sexing technology. So other differences are also will be there, not only DNA content, other differences also will be there. Size of the X chromosome will be high than Y chromosome, and the surface charges of the sperm also. So X chromosome bearing sperms will be having a different electrical charge than Y chromosome bearing sperms, and cell surface antigens also. So the sperms will be having antigens on the plasma membrane. So the antigens also will be different in sperms having X chromosome and sperms having Y chromosome. So in this way, the both type of sperms will be having different types of cell surface antigens. They will be having different types of surface electrical charges, and they will be having differences in size, and they will be having differences in terms of DNA content. So these are the basis for separating these sperms into X and Y chromosomes. So, and what is the use of this sperm sexing technology? So offspring of choice. So we don't want it, we don't want to leave this to choice. We want to decide whether we want to have a female offspring or male offspring. Basically, this sperm sexing technology has been devised for using in dairy technology only. So in dairy technology, usually we use female cattle for milk production, and male organisms are like bulls will be used for meat production only. So cows, they will give more amount of milk, but they are useless if we want to establish a meat production industry. So if the individual is looking for meat production, then he should have males only, he should have bulls only. And if the person is desired to produce milk only, he is not going to produce meat, or he is not going to sell meat. So he is going to sell the milk only. Then rearing or growing these males is useless to him. So he requires only female cattle. So for that purpose, for different needs, we would like to have different gender cattle. So it should be in our hand. So if the breeder, let us assume the breeder is a milk producer, while he is breeding the cattle, almost 50% of offspring will be males and 50% offspring will be females. But females only are useful to him, males are useless. So, but he cannot kill them. He cannot, what he will do with those males? So for that purpose, before males are born, it would be better if he has a choice that all the born offspring are females only. So in such scenario, this sperm sexing technology becomes handy. And breeding stocks, not only this cattle, remaining all cases also, breeding stocks also, the breeders will be different. So usually dairy people will be different, and meat producers also will be different. And the breeders, they will produce either male or female offspring, and they will sell these offspring to the dairy people or meat producers. So for that purpose, breeders also, depending on their needs, depending on the demand, they would like to go for this many number of female offspring are required, and this much number of male offsprings are required. So their requirements will be different. Depending on their requirement, they would like to have the choice of offspring in terms of gender. So this X sperm contains usually 4% more DNA than Y sperm. This is the basis, main basis for this sperm sexing technology. And different types of methods have been developed by the scientists. Out of them, let us see a few important ones. First one is gradient swim down procedure. In this gradient swim down procedure, it will be done in test tubes. A liquid, a solution will be taken, and in this solution sugar will be there, and albumin gradient also will be there in this thing. So usually Y sperms are smaller in size than X sperms. So while these sperms are going for this sugar solution, why? Because they require energy. So to survive, these sperms require energy, and energy usually will be provided to the all cells by sugars only. So they will be going for sugars, so for taking sugars. So when sperms are added to this solution, which is having sugar, the Y sperms will go fastly than the X sperms, so that we will get more energetic Y sperms in the bottom of the test tube, and less energetic X bearing sperms will be on the supernatant. So in this way, here a clear gradient will be formed in the test tube. So fastly swimming Y chromosome bearing sperms or Y sperms go deep than the X bearing sperms. Why? Because these are less energetic. So in this way, based on this thing, a different difference is there in between these two types of sperms, that is called as gradient swim down procedure. And second method is surface antigenic differences. And these two types of sperms, X sperms and Y sperms, they will be having different types of antigens in their surfaces. And here, in case of Y sperms, a HY antigen will be used as a marker. And apart from these HY antigen, sex-specific proteins also will be there on the surface of these sperms. And non-SSPs are removed immunologically. So apart from this, along with these SSPs, sex-specific proteins, some non-sex-specific proteins also will be there on the plasma membrane of both sperms. So these non-SSPs will be removed immunologically. So by adding them to corresponding receptors, those remaining non-SSPs will be removed. Only these SSPs, sex-specific proteins, that means special antigens only will be there. So only those antigens which are present on Y chromosomes will be there, and those only proteins that are present on Y chromosomes only will be there. Remaining commonly occurring proteins will be removed. So now we can separately choose these sperms. So by adding the corresponding markers, we can bind the required sperms only. So in this way, usually we know that in immunology, a specific antigen binds with a specific antibody only. Okay, so a single antibody cannot bind with all antigens. So antigen and antibody are specific. Okay, let us assume some X antigen is there, it will bind with the X antibody only. So here in this case, we are having two types of antigens. One is Y antigen, another one is X antigen. So remaining are common antigen. Common antigens will be removed. So after removing the common antigens, now sperms are having only sex-specific antigens. So now if we want to have only Y chromosome sperms, then what we will do, we will add the antibodies which can bind with Y antigen only. So after adding these antibodies, only Y antigen binding sperms only will be bind to added antibodies. Remaining sperms will be having X antigen and they will not bind with the antibody. So that what will happen when we wash the liquid, only Y chromosome bearing sperms will be attached to the antibodies. Remaining X chromosome sperms will be washed away. So in that way, we can discard X chromosome bearing sperms and we can retain only Y chromosome bearing sperms. If we want to have the if we want to have the X sperms, then we will add X antibodies. So in this way, this is just like a basic immunological method. So by applying this basic immunological method also, we can separate or we can get required sperms from the collection of the semen. So next, third method is the free flow electrophoresis. So in electrophoresis, you might have studied about gel electrophoresis method in first year. So in electrophoresis, what we will do, we will separate the chemicals from a sample depending on their differences in electrical charges and depending on their differences in sizes or molecular weights and many other factors are useful in separating the chemicals from a sample. So, but there we will use electricity power. So electrical energy will be used to separate these molecules from the sample. Here also we can use this electrophoresis method to separate the X sperms from Y sperms. So why? Because both these two sperms will be having different surface charges on them. So we also add some chemicals to create some differences in their surface electrical charges. So after doing this thing, we will allow this to we will subject it to this total electrophoresis method. So by allowing it to undergo this electrophoresis method, we can separate these two types of sperms into two major bands on the agar gel or whatever gel we will take. So from the medium later, we can separately collect these sperms from these two bands. So this is just like a electrophoretic method. And last and fourth important one is volumetric differences. In this volumetric differences, X sperm is larger than Y sperm. X sperm will be having more DNA than Y sperm. So this difference is very useful in separating them. Apart from this size and content, there are differences in sperm head volume also. So Y sperms and X sperms will be having differences in the sperm head. And here in this thing, a microscopic method and image analysis, we use in ultrasound scanning, we use monitors. So all those monitors also will be utilized, and flow cytometers also will be used. So by combining all these microscopic method, image analysis methods, and flow cytometer methods, all these things are utilized to separate these X and Y chromosomes. So example for this volumetric differences method, flow cytometer is a better method, and this was the first method devised by this American government. So the basis for this flow cytometry is that, as we have already told, the differences in DNA content is the basis for this flow cytometric method. In this flow cytometric method, we use this imaging devices like computer monitors, and we also use some electrical differences, and this total thing is called as a flow cytometer. So this apparatus is flow cytometer. So flow cytometer will be utilized, and the imaging technology also will be utilized in this method. So in how it will be done. So first we will take the sample, semen sample or sperm sample. To the sperm sample, we will add two types of dyes or coloring agents. Out of these two, one is Hochet 33342, and second one is red quencher food coloring dye. So this Hochet 33342, it will bind to the DNA only. So it will bind to the chromosomes and DNA only. And that too particularly sex chromosomes, X and Y chromosomes only will receive this Hochest 33342 dye. And this red quencher food dye, it will be received by dead and non-intact sperm membranes. Some sperms will be dead, some sperms will be non-motile, or some sperms will be inactive in the sample. So such type of useless sperms will be receiving this red quencher food coloring dye. So like this, two types of dyes will be added. Out of these two types of dyes, red quencher dye will be received by useless sperms, and X and Y chromosomes will receive Hochest 33342 dye. And depending on the DNA content, X chromosome will receive more Hochest 33342 dye than Y chromosome. Why? Because Y chromosome is having less DNA content only. So after this thing, what we'll do, the sample will be taken into this flow cytometric method. Here in this, a vibrator will be there. So when it is started, the apparatus is started, the vibrator will start sending the sample like a drop by drop. So, and all these drops will travel through a nozzle. And when the semen drops are going to fall in downward direction, here we will arrange a laser machine. So from this laser beams will be emitted, and these laser beams will be received. The laser will be received by detectors. So two detectors will be arranged in this machine. One detector will be arranged in 90 degrees angle to the laser, and another detector will be arranged in exactly in opposite direction, that is at 0 degrees. So the forward scatter detector will be arranged in 0 degree direction. So it is in forward direction. And another one is, this is side scatter detector. So if the sperms are scattered in sideways, then that will be detected by this 90% or sideways detector. And if the sperms are going straightly, then they will be detected by this forward scatter detector. So this forward scatter detector will be utilized for knowing that, identifying amount of DNA. Total amount of DNA will be identified by this 0% detector, 0 degree detector, and fluorescent intensity. Fluorescence intensity means the Hochest 33342 dye will give fluorescence. A type of emitting light will be one type of fluorescent light will be emitted from this X and Y chromosome. So the amount of emitted light will be measured by this 90 degree detector. So in this way, both these two things are detected by this 90 degree detector and 0 degree detector. And below this in this apparatus, there will be some deflector plates will be there. These deflector plates will be having separate or different charges. One plate will be having positive charge, another plate will be having negative charge. So these two things are arranged in opposite directions, and three vessels will be three bottles will be placed under these total apparatus. One will be placed under this positive charged plate, another will be placed just below the negatively charged plate, and one will be placed exactly in the middle of the total this water droppings. So when the drops are falling in between these drops, in between these deflection plates, depending on their charges. They will either they will be attracted towards positively charged deflection plate, or either they will be attracted towards negatively charged deflection plate. X and Y chromosomes only will be attracted towards positive and negative charged plates. And the dead or useless cells, useless sperms, they will not be attracted towards these cells. So all those waste materials will be collected exactly in the middle of this, in the middle vessel or in the middle plate. And X chromosomes, usually they will be having negative charge, they will be attracted towards positive plates, and Y chromosomes will be having positive charge, they will be attracted towards this negative plate. So if we want to have only X sperm, we can place only one bottle or one plate under this positively deflection plate. Or if we want to have only X sperm, then we can place the vessel under this negatively charged deflection plate. So in this way, by operating the total machine in this way, with the help of these differences in this DNA content, and DNA content also, the DNA content also will be will allow us to differentiate the chromosomes into, or sperms into X and Y, depending on the differences in their DNA content. And both these two things are also will be, both these two X and Y sperms can be collected separately. So this is the way the flow cytometer method is operated, and we can collect the sperms separately. So after the sperms are collected separately, then they will be subjected to purity tests. So purity analysis or sorting analysis, sorting reanalysis will be done by observing them under the microscopes. So after assuring that good quality sperms are collected, then they will be used for this storing them, and for storing cryopreservation method also will be utilized. So with the help of this cryopreservation method, we can preserve the method for many days under very low temperatures. So, and what are the advantages of this flow cytometry or sex sorting technology, or sperm sexing technology, is that it increases the reproductive rates in animals, or usually in cattle, and subsequently it also increases the rate of genetic gain, and positive the higher selection intensity also will be made possible with the help of these methods. And this is very useful in dairy industry, and animal breeders also, they will are very benefited from this technology. And it is economically also benefit to the dairy farmers, because they need not to unnecessarily feed the male cattle. So feeding the male cattle is not economical for them, so that economically also these dairy farmers will be saved a lot. So these are the advantages of the sex sorting technology, or sperm sexing technology. And what are the limitations? So there are some limitations also in this methods. This is very slow process, and half of the sperms are unsexable and go waste. So this is not actually this, we cannot call it as a limitation. So actually we don't want them, so that half of the sperms will be discarded, and only 30% of the sperm are sexable or can be separated. And out of that 30%, only 15% is responsible for female offspring. So success rate is very low. So even though we collect a lot of sample, finally we will get only limited amount of required sperms only. And cost of this total experiments or procedures is also very high. And due to this addition of these chemicals or dyes and applying these electrical impulses or electrical charges. So due to all these methods, the sperms also may get damages. So they genetically, they may get some damages, and these damages will reflect in the offspring also. And conception rates are also very, very low with the usage of this sexual semen. And for doing all these things, skilled manpower is also, so skilled persons are required. So normal people cannot do this sperm sexing technology. And choosing one sex over other can affect the normal Hardy-Weinberg equilibrium in the population. So after some days, what may happen, only the female population will be dominated all over the world. So male cattle number may be reduced, and after some generations, there may be a chance of the total this procedure may become as a tool to the extinction of the species itself. So in this way, deciding or controlling the only one sex, so controlling, having a control in our hands is not natural. So that is also another ethical objection ethical objection raised by some other persons. So these are the limitations of this sperm sexing technology. So with this, topic is over.